Hey, good afternoon, everyone. I'm Bob Thacker. I'm the chair of the Department of Ecology and Evolution. And it's my pleasure to welcome you to the third presentation of the Rohlf Medal for Excellence in Morphometric Methods and Applications. The Rohlf Medal was established in 2006 by the family and friends of Dr. James Rohlf to celebrate his 70th birthday. And Dr. Rohlf is currently an emeritus distinguished professor in the Department of Ecology and Evolution and a research professor in the Department of Anthropology. Recipients of the Rohlf Medal are recognized for excellence in their work on the development of new morphometric methods and applications, and these include the biomedical sciences, including evolutionary biology, population biology, physical anthropology, and medicine. So today, it's my pleasure to welcome you here for this lecture and to introduce Dr. James Rohlf.
I'd like to first thank the members of the selection committee. They consist of Paul O'Higgins, who is there. He'll act as the moderator for the lecture. He's center of the head of human paleoecology, evolutionary origins, and professor of, foundation professor of anatomy, the Hull York Medical School. He also served as chairman of the selection committee this year. Fred Bookstein, there, is a retired professor of morphometrics. I think the only such title in the world, a professor of morphometrics. University of Vienna. He's also in Department of Statistics at University of Washington. And he was the first recipient of the medal. Dennis Slice, there. He's a professor in the Department of Scientific Computing, Florida State University. He's also a graduate of the Ecology and Evolution Department. Eric Delson couldn't make it. He's professor of anthropology in Lehman College of CUNY and also director of the New York Consortium of Primate Anthropology, Evolutionary Primatology. He's also a research associate at the American Museum. Bill Youngers was also part of the committee via virtual connection with Skype. He can't be here because he's in Madagascar. Mike Bell, also of this department. Do I see Mike? He served as sort of helper and advisor on local arrangements. Okay, for this year's award is to Ben Hallgrímsson. He's being recognized for this medal because he has been at the forefront of efforts to apply morphometrics to the field of evolutionary developmental biology as well as anthropology. He was the first to apply geometric morphometrics to the study of prenatal shape variation and growth. His studies combine the quantitative power and detailed anatomical characteristics of phenotypic effects provided by morphometric methods with the knowledge of developmental processes that are responsible for those effects. He has collected morphometric data by micro-CT or optical projection tomography, pioneered the application of geometric morphometrics to large samples of embryos through extensive collaborations across boundaries between developmental biology, computer vision, bioengineering, and has relevance to medicine as well as the science of developmental biology and the field of morphometrics. He's brought these methods to new audiences where they continue to make an impact. If geometric morphometrics becomes widely accepted as a technique in developmental biology, this will be largely due to his work. Ben? This will be done in 2 stages. I'll give him the medal, then he has to step back. Fred will introduce his lecture.
Okay, I thought I was supposed to start talking.
And I give you advice that Fred found out right away. You won't want to wear this the whole time. It's surprisingly heavy.
It looks quite substantial.
But I can present it to you. Get some Velcro to stick.
Okay.
There.
Thank you very much. Thank you.
Congratulations.
Thank you.
Oh, good afternoon. Benedikt Hallgrímsson, our beribboned friend here, is a friend of mine. I've published a little as his co-author and a lot as contributor to the major journal, Evolutionary Biology, that he edits. I've written grant proposals with him to the Canadian Federal Medical Science Foundation, unfunded, alas. I picked him as one core speaker at the meeting on measurement in biology that I ran a few years ago in suburban Vienna, and I've frozen with him. Well, he didn't freeze. He was properly dressed, walking through downtown Calgary on a random evening at minus 10 degrees Fahrenheit during a winter visit. I tried to get him to apply for the chairmanship of my department in Vienna, in other words, to be my boss there, but wisely he declined. Ben has even fed me steaks from his father-in-law's ranch. So yes, he's a friend as well as a colleague. In view of how different we are, this sort of friendship is, on its own terms, another good reason for admiring his professional achievements. Ben spends a lot of his time with people whose training is a long way from his. Me, I'm a mathematical statistician. Trained in anthropology, he now works and publishes in evo-devo, birth defects, medical imaging, genetics of signaling pathways, craniofacial biology, morphometrics, theoretical biology, and, of course, anthropology. He's integrated with the bioengineers on his campus and likewise with the computational imaging folks who share his concerns about problems like the locating of landmark points in samples of a couple thousand skulls imaged in parallel. It's hard work to learn to speak these languages, and Ben has put in the hard time to learn them successfully. As I look back on Ben's career so far, with 20 or 30 years still to go, the main theme I see running through its diversity is that after his early work on fluctuating asymmetry of scalar measures, he gambled and won on the potential of the new morphometrics, our geometric morphometrics, to have an immediate and transforming effect on experimental results and reports in his main laboratory field, which is the craniofacial impact of genetic knockouts. Beginning about 10 years ago, he and his colleagues produced a protocol for data handling and reporting in experiments of this sort that has by now been canonized in textbooks, workshop presentations, and, just to pick a current example, in the master's thesis of my most recent master's candidate at the University of Vienna. This goal works both ways. Whenever a new morphometric statistic comes along, like Philip Mitteracker's 2009 suggestion of a distance measure among multiple covariance matrices, Ben will publish it as soon as it passes review at his journal. Immediately afterwards, apply it himself to some data lying around, and add it to the toolkit thereby that he displays for others to imitate. This year, he treated my suggested new approach to the classic topic of integration the same way, and my radical rebuild of the Procrustes toolkit is on his desk right now. In that way, Ben acts as a filter for the best new tools. And this brings me to the crux of the matter, which is the way he represents, I think, the future of morphometrics. Benedikt Hallgrímsson is a perfect example of what our interdisciplinary community is trying to evolve into. A network of people who reflect and refract their biological imagination in order to serve all the different social systems, research, teaching, academic administration, that are involved in sustaining our professions. I know only one other prominent researcher into human biology who was disciplined enough to embrace this sort of multiple altruisms, Robert Moyers, my mentor at Michigan 40 years ago. And he was more a craniofacial biologist than an anthropologist. In Ben's career, I see the same energy that drives the best academic intellectuals anywhere in the sciences, a concern not just for the facts and findings, but also, and perhaps primarily, for the maintenance and perpetuation of the institution that makes this life possible. He is, after all, not only a professor and research scientist, but also an associate dean of medicine, specializing in education. One referee for the Rohlf medal mentioned that Hallgrímsson has a habit of supplying cover photos for the journals in which he publishes. That is telling. His concern for the dissemination of the new evo-devo science to the human groups that, by their collective legislation, make it possible to continue the work. As I see it, He represents the most important future trend for morphometrics in its medical context, the outreach to all the other scientific communities that deal with the human beings of whom the images are, or eventually will be, the primary subject of discussion, and to the publics of those scientific communities. In other words, Ben's career furthers the overarching goal I've always associated with contemporary academic life at its best, the commitment not just to new science, but also to the community of people of every generation who appreciate that science, who themselves might occasionally, or even more than occasionally, meditate on or wonder at biology or medicine for the way it can illuminate the essence of our species and our self-awareness of it. I can't think of anybody inside or outside of morphometrics or anthropology who illustrates this better than Benedikt Hallgrímsson. The final sentence of his Rohlf Award letter reads, "For his excellence in evo-devo morphometric research, for his advances in the imaging technology that drives the research, and for his varied and energetic efforts at disseminating all this to a broad variety of audiences, Benedikt Hallgrímsson richly deserves the honor of this third Rohlf Medal for Excellence in Morphometrics." Yes, indeed, this award is richly deserved. Ben has chosen to speak today on the middle-out approach to complex traits, morphometrics and the middle-out approach to complex traits. Please join me in welcoming him to this podium for this third Rohlf Lecture Prize on Morphometrics. Ben?
Well, thank you very much, Fred. That was an amazingly generous introduction, and I'll see if I can actually remember my talk now after all that.
I didn't use any slides.
And I should say also that I'm deeply honored to be the recipient of the Rohlf Medal. And I also, as I was telling Paul earlier today, I feel a bit sheepish about it because there are so many people in the field who I think are deserving of recognition and whom I probably would have picked ahead of me. And so, and I feel like I often use methods that have been developed by others, and I'm a consumer of the good work that is being done in the field. So I think on behalf of, I'd like to thank all of those people for their contributions, because because what I have done in terms of bringing morphometrics into developmental biology would certainly not have been possible without the work of so many others, and of course not without the foundation that was built by Jim and Fred and some of the key people in the field. So thank you very, very much. Okay, so let's see if I can remember the talk after that. Actually, I think, Jim, you're right. I think you need to take this off before. It is a medal of significant heft. So my talk today is, can you all hear me? Okay, is about the middle-out approach to understanding complex traits and the way in which we use morphometrics to do that. So we basically work on the developmental genetics of complex traits. And we do this really for two reasons. One is that we're interested in the evolvability of morphology, so how selection produces changes in developmental processes that in turn drive morphological variation over time. And we argue, of course, that understanding the developmental basis for this change, for morphological change, is critical. We also study complex traits in the context of structural birth defects, and particularly structural birth defects of the face. And the reason that we're interested in the developmental basis for structural birth defects is because we're interested in the generation of variation. And this is a topic that I think is increasingly becoming linked to this broader topic of precision medicine or individualized medicine, which I think, certainly as an evolutionary biologist in part, that precision medicine really is about variation. It's about understanding why patients are different from each other and how the things that make them different from each other make them respond differently to treatments or have different types of prognoses. And I think that realization that precision medicine is all about variation is one that's increasingly becoming clear within the broader biomedical community. And the particular complex trait or complex of complex traits that we study is the shape of the skull and the shape of the face, so the craniofacial complex. And I think, as you can all, if you know anything about about the skull and the face, you know that this is a highly complex, highly integrated, highly variable trait, and of course, one of great interest. The problem with complex traits, of course, is that they're complex. And they're complex in ways that I think are somewhat obvious. So those of you who may work on the genetics of these traits, of course, are familiar with the fact that polygenic traits often have genotype-phenotype maps that would look something like this abstraction, where there are where multiple genes affect a trait and where individual genes can affect multiple traits. So there's essentially a many-to-many relationship between genetic variation and phenotypic variation for complex traits. And this actually makes it complex enough because with all of the possible genetic variants you have, and current genome-wide association studies are often looking at, say, 60 million SNPs in one go, we don't have the money, the sample sizes, the wherewithal really to test the individual effects of all the different possible SNPs on phenotypic variation. And so the approach that genomics is currently taking, or is currently, I guess you could say, trapped in a way is impossible. And it's impossible because of the complexity of complex traits, even if you just look at them with this sort of simple genotype-phenotype map abstraction. But of course, we know that the genotype-phenotype map is much more complex than that. And it's more complex than that because genetic variation plays through developmental processes. Developmental processes act at different levels and times in development. They interact with each other. They have an environmental context or environmental influences that then in turn act on these developmental processes. And so this is the way in which genetic variation flows through to phenotypic variation. It's via all this stuff here in the middle. And so what I'm going to argue in this talk is in part that understanding the stuff in the middle is important, because without it, you really can't understand how variation is generated. But it's also an opportunity, because in many ways, the stuff in the middle simplifies what we can get from, or what we can derive as an understanding of the basis for variation in complex traits. So here's a typical complex trait. It's one that is one about which we probably know more than almost any other. So this is human stature. And I picked this example just because it illustrates very nicely what I mean by this complexity. So stature has been the subject of multiple genome-wide association studies. And this is a recent excellent paper that, of course, I was not involved with, but it's a very good paper that put together the results of genome-wide association studies in recent years for stature. And they amassed a sample of about 250,000 people that were fully genotyped. And they arrived at about 697 SNPs that reached genome-wide significance in this massive sample of 250,000 individuals. And more than that, though, they did a whole bunch of really clever secondary analysis, including estimating the number of SNPs that should be there based on what they found, that would explain the full set of variation in stature. And of course, you know stature is heritable. It has a heritability of about 0.8. And they estimated that 9,500 SNPs would explain about 30% of the variance of stature. These 9,500 SNPs probably distributed across a significant portion of the genome. So this kind of begs the question, well, if the final number of SNPs, for explaining all 80% of the variance is even higher than that, maybe 20,000 or something, what does that actually tell you? What does that tell you beyond just saying that stature is heritable, that genetic variation contributes to stature? And I would submit very little. There's very little useful in a genetic explanation of stature if it's that complicated. And that's one of the reasons to try to look for different approaches. Now, I'll come back to this at the end, but they did another very clever thing, which is that they took those 697 SNPs that reached genome-wide significance, and they did a network analysis, and they found out that a significant proportion of those genes, in fact, they were one of these nodes, I think it's this one over here, one of the nodes that they defined within the network had to do with cartilage development and growth. Seems reasonable enough. But that actually tells you something useful about the genetics of stature. It tells you, that gives you a developmental locus to focus on that gives you hypotheses as to what is causing variation in stature. Okay. So the work that we have done in this area has really focused on two central phenomena or central ways in which development structures phenotypic variation. So we're interested in the structure of phenotypic variation. And I'm going to talk a bit about these two phenomena and relate them, well, actually, I relate them at the end, both to complex traits in general and also to evolvability. But we worked on this phenomenon of how developmental processes integrate variation into patterns of covariation. So this is morphological integration. And we've also worked on the phenomenon of how developmental processes modulate the amount of phenotypic variation that you get expressed, either within genotype, within individual, or relative to genetic variation. So this is the phenomenon of robustness, or the extent to which development produces predictable phenotypes in the face of perturbations. So let's start with integration. So this is the first part of the talk, about 2/3 or so of it. So integration is simply, it's a reflection of the underlying structure of developmental interactions. And it is the cause of covariation among traits. And so what I mean by covariation among traits is that you have some source of variation, some perturbation, and you get a set of correlated effects in the phenotype. And one way in which we see the effects of integration very clearly is in the case of human syndromes. So here's the case of achondroplasia, and here's Peter Dinklage, who's my favorite actor from the Game of Thrones, a wonderful actor, and I should say a very handsome guy. But he does actually exhibit these typical features of achondroplasia, which is that you have a bossing of the forehead, short, you see it better here, a short chondrocranium, and a reduction in midfacial prognatism, and also a change in the cranial base angle. So these are all correlated features of achondroplasia that, in a sense, represent the manifestation of integration. And this kind of structure is a general feature of most morphological variation. And you can see this here in this analysis of variation in the human face. So this is an analysis of about 6,000 Tanzanian kids. And here we've just done a principal components analysis. And these are the first 10 principal components here. And the first 10 principal components in this data set account for an overwhelming majority of the variation in this data set, which speaks to a moderate level of integration. Although compared to most other morphologies that we work with, including things like the mouse skull, the human face actually is not all that integrated, or does not exhibit all that of a strong covariation structure. And if you look at so this is from a study that I've been involved with now for many years with Rich Spritz at the University of Colorado, Denver. It's a genome-wide association study of human facial shape. And so because this is a GWAS study, we also have genetic data for these individuals and these traits, and we can calculate a genetic correlation matrix, which Joanne Cole in Rich's lab has done. And you can see a reflection of underlying structure also in the genetic correlations among the traits, and also in the patterns of heritabilities among the principal components and also various traits. So that's an example of structured variation. So here's another example. So I worked recently with Axel Bissell on the effects of enhancers to craniofacial genes on craniofacial morphology. And when Axel contacted me initially about the study, he was very particular to tell me exactly when and where the mutations that he was interested in, or most interested in, were expressed, because he expected to find highly localized effects. And I told him immediately, actually, that even though these are very subtle effects, that's not what you expect to find, because these effects are all acting early in development, and you would expect to see them cascading through development, through these various levels of interaction. And what you get in the end is some sort of covariant, or some sort of pattern of covariant effects that we predict, or I predicted rather, that these enhancer effects would have integrated patterns of effect, and of course they do. So they don't just produce an effect in one localized area, they produce an effect that is distributed throughout the skull. So here's another example of structure and variation, and this time somewhat unexpected structure. So this is work that I've been doing with Michaela Pavlichev and Philip Mideroker and others. So this is the case of structure in the dominance deviations in the sets of crosses between the founder strains of the collaborative cross. We have 8 founder strains, and we cross them all, and we got the 64 minus 2 that didn't work, possible combinations of the 8 founder strains, and then you can localize the expected place where you expect to see each of these crosses, right between the 2 parental strains, and you can then plot where the actual strains fall relative to where they were expected to fall. And what you find in that, I know it's a bit hard to see in this, but you do find non-random directionality in the dominance deviations. And this is even after you've taken out a Llama tree. And so this is, for example, the pattern of shape change in PC1 and PC2, an analysis that Philip Midderoker did. And the direction of departures from the expected mid-parental value is non-random in direction. So that's an unusual thing, because these perturbations are coming from all over the place. Why would the dominance deviations in a set of crosses be non-random in direction unless they are playing through a highly structured set of developmental processes, that there's a non-random effect whereby genetic perturbations tend to be shunted in particular ways because of the way in which they're flowing through development. So here's a case of unexpected structure in development. So how does this kind of structure of variation arise? In other words, how does covariation arise in development? Well, covariation, as I said earlier, is the consequence of integration, and I want to be clear that it's not to confound integration and covariation structure. Integration is a set of connections that enables covariation to arise. A population of clones living or raised under identical conditions is just as integrated as a population in which there's standing variation, but it will exhibit no covariation because the connections can't be realized into covariation if there's no variation. So covariation arises in development because you have variation in something, such as the expression of a gene, like FGF8 here, and that gene will affect some developmental process, like mesenchymal proliferation, as you can see here in the facial prominences. And that process, with variation in that process, will in turn produce covariation among the embryological structures that have that developmental origin, which in turn produces covariation among the later developmental or adult structures that share this particular developmental origin. So covariation arises from variation in developmental processes. It's from the integrated effects of variation in developmental processes. Covariation structure arises from integration. Now, what's difficult about this is that for most traits, like the shape of the skull, there are many developmental processes that can vary and produce integrated effects, can produce patterns of covariation. So you can think, if you think about the list of potential processes acting at different times and partially overriding each other, you can see how in natural populations, covariation structure is basically indecipherable from a developmental point of view. And I think there may well be some exceptions to that, but I think it's probably generally true. So you're probably wasting your time if you're trying to make inferences about development from covariation patterns in natural populations. And this is the phenomenon that we call the Palimpsest. And the important thing about the Palimpsest model for integration is that you recognize that integration arises from covariation acting at different times in development, that these processes can override each other, can interact, can produce unexpected effects. And it's the sum total of all these processes that produces the covariation structure that you end up with in the end. Okay, so the approach that we take have taken over the past few years then is to, instead of trying to dissect this stuff in natural populations, is to use a perturbation approach where we take particular developmental processes and we construct hypotheses about how they might affect development, and then see if we're right by using mostly mouse models, or in some cases chick models. Okay. So I'm going to tell you 3 short stories about integration. And one of them, the first one, I think represents a significant, or several years' worth of work, and it's work mainly that my group has done in collaboration with Rohlf Marcuccio's group. Rohlf and I have been working together for several years in this general area. And that's the regulation of the formation, outgrowth of the midface, and in particular, the way in which the development of the brain and the development of the midface interact. So before Rohlf and I started working together, he had discovered this important signaling center in the face, which he named the FEZ, or the frontal nasal ectodermal zone, and it's an expression domain of sonic hedgehog that you see here in the ectodermal epithelium of the face. It's set in motion by sonic hedgehog expression in the forebrain here. And together, sonic hedgehog expression here and here regulate proliferation in the intervening neural crest-derived mesenchyme, which then produces outgrowth of the face. So Rohlf and I started working together because Nathan Young, who had done his first postdoc in my lab, went to Rohlf's lab, and then I met Rohlf through Nathan, and Rohlf and I hit it off because we both like to go to nice restaurants and eat really good food and cook food, and so discussions about food and wine led to discussions about developmental biology and integrating morphometrics and developmental biology, and I should say that Rohlf has also been very instrumental in pushing that integration within that field. And so quickly we latched onto this topic that very little was known about the generation of variation, about the mechanisms that actually produce continuous variation in complex morphological traits. In fact, no experimental system had ever been devised to our knowledge that modulated up and down a process in which you get continuous variation in a complex morphology. And so that's what we set out to do. And so the design of the experiment was that we took this system that Rohlf had worked out and changed the amount of sonic hedgehog expression that you get by putting either by putting sonic hedgehog into the brain or repressing sonic hedgehog expression in this part of the brain. And that would then, we hypothesized, alter the fez and the expression of sonic hedgehog in the fez, and would alter the shape of the face. And this is, to summarize a lot of work, is pretty much what we found. So when you change the amount of sonic hedgehog up and down, this is the wild type range here. So this is down regulation of sonic hedgehog and up regulation here. You get this range of variation in the overall shape of the face. You also get, or you can also relate rather the rate of cell proliferation in the midface to the overall shape. And you can also relate the sonic hedgehog expression directly to facial shape here within this upper range. And what's interesting about this range of variation is that it's very broad. So it actually extends the entire range from something that would look like holoprosencephaly here to something that looks like mid-facial clefting here. And of course, if you know anything about holoprosencephaly, you know that the sonic hedgehog pathway is implicated in holoprosencephaly in human patients. So this is not to say that sonic hedgehog is a major driver of facial shape variation. In fact, it doesn't seem to come up very much in the human GWAS studies of facial shape. But it is to say that this pathway and the mechanisms by which that pathway is modulated may well be an important central determinant of this particular aspect of facial shape variation, which is, of course, the width of the face. So that would be one place to look for some regularity in this complexity. Most recently, Rohlf and I have gone on with the help of Diane Hu, who's a wonder technician in Rohlf's lab. She has magic hands, and if you do developmental biology, you know the importance of this. So her magic hands are able to do this incredible experiment where she's able to take half of a brain from one embryo and transplant it to another embryo, and then induce or get that brain to be incorporated. And of course, if that brain is expressing some genes, it's going to produce an effect, produce some sort of change. And so this is exactly what we did. So Diane took hemibrains from chicks and transplanted them into quail. And she took hemibrains from ducks rather, and transplanted them into chicks. We did a variety of different versions of this experiment, but this is the one I'm showing here. And so what we predicted was that you would, by transplanting the hemibrains from the ducks, you would change the shape of the face, change the change of the sonic hedgehog expression domain, and in fact, that's exactly what we showed. So this is the sonic hedgehog expression domain here. When we started to do this, we found out, though, that it was really difficult to quantify the shape of these sonic hedgehog expression domains. And initially, we actually spent quite a lot of time trying to use geometric morphometrics to put landmarks on these fuzzy shapes, and it wasn't working very well. Although actually James Sharp has done that in a different context. But we couldn't get that to work here. And so I was lucky enough to meet at a conference Washington Mio, who is a mathematical statistician at FSU. And he had this method called spherical harmonics analysis, by which he could directly obtain shape information from the shape of a three-dimensional object using the predicted frequencies at which that object would vibrate. And there's a one-to-one correspondence between those axes and the overall shape of the object. So here are the first two principal components for shape variation in the fez, and you can see that actually on this plot the chick fezzes and the duck fezzes separate very nicely. And when you do morphometric analysis, both of the chick and duck fezzes and also the duck chick and chick duck fezzes, you end up with an interesting pattern. Let me just show you what it looks like here on these pictures. So here's a chick, and here's a duck. And notice that the nasal pits are differently shaped in the chick and the duck, and also that the fez has a different shape in the chick and the duck. It's sort of more triangular in the chick, and the duck is a bit more rounded. It's differently shaped. And here's a chick-chick control individual. So this animal was operated, but a chick brain was taken out and then put back in. And here is a duck-chick chimera. Actually, these labels should be reversed because this is the duck side and this is the chick side. And see here how the nasal pit on the chick side is more chick-like, and the nasal pit on the duck side is more duck-like. It looks more like a duck than it does like a chick. And the same thing with the fez. Look how the fez is sort of rounded on the duck side, and it's more chick-shaped on the chick side. And there were also similar changes in the eye that correspond to this. And when you quantify both facial shape and the shape of the fez, you end up with a partial result, but it's a result that shows very clearly that the duck-chick transplants, the hemifaces, move in the direction of the shape of a duck. So you induce basically a partial transformation of the shape of a chick face into a duck face by transplanting the brain. So what this means then is that the fez and the sonic hedgehog expression of the fez must be a very important determinant of the overall shape of the face, because if you transplant the fez from one species to another, you get a partial transformation. Okay, so let's move up one level then in this interaction between the brain and the face. As the face forms, it basically grows on this platform of a growing brain. So it forms on this platform of a growing brain. So there are mechanical ways, in addition to signaling pathway ways, in which the brain and the face interact. And these mechanical forms of interaction actually start quite early. So they start, for example, during the formation of the face. So you can imagine that as the face forms from these facial prominences that have to come together, that they need to grow at a certain rate and in a certain direction to overcome the fact that the brain is sort of growing and basically pulling the face apart. And that actually turns out to be exactly the case. So if you have mouse mutants that reduce the rate of growth of the facial prominences, which we show here, and I won't go into the details, you end up with an altered shape, and it's exactly the shape that you would predict if the brain were sort of stretching out the face. So there's a shape interaction effect that results from how the face is forming on this growing brain. And later in development, the brain continues to be important for craniofacial shape. And in anthropology, there's this long-held idea, which I think really actually comes from De Beer, but was formalized by Biegert in the 1960s, that tried to explain the particular form of the human face, where the human mid-face is sort of tucked in beneath the brain as a consequence of growth of the brain with a relatively small face. And so what Biegert argued is that as the brain grows, it basically flexes the chondrocranium and produces this effect where the face is sort of tucked in underneath the face. And Dan Lieberman and I have worked on this in mouse models, and this is just an elaboration of Biegert's model in 3 dimensions, in which we tried to figure out how that physical interaction might actually work in 3 dimensions. And so we hypothesized that as the brain gets bigger, the mid-face is going to get pulled in, and you're going to get an increased flexion here of the cranial base angle. And when you test this out in mice, this is exactly what you find. So these are MSEF mice that have overgrowth, generalized overgrowth of the brain. And as the brain gets bigger, the cranial base also gets more flexed, the neurocranium gets more domed, and the face gets pulled in, of course, to a much lesser extent than in humans. But this is exactly the direction of change that you would predict based on Biegert's model. And what it tells you is that there are higher level mechanical interactions that are happening during growth and development between the face and the brain. Okay. Oddly enough, even though humans have these huge brains, when we've tried to see how much variation you would explain from the size of the cranial base in humans, and this is work with Neres Martinez, it actually doesn't explain that much. It's about 1 1/3% of the total variance of the shape of the head. And mice, which have much smaller skulls and obviously much smaller brains, although they're still relatively large compared to the size of the skull because mice are small, the size of the brain explained more variation in a sample of outbred mice. Okay. You can't actually see this OPT image. So the next example that I'll give you is cartilage growth and craniofacial shape. And so I put up these pictures to show you that, of course, you have the skeletal system forms from two kinds of bone that goes through a cartilaginous intermediary step, and bone that ossifies directly from intermembranous bone. And the thing about the skull, which is shown here, this is an OPT image of a neonatal skull, is that you have both of these kinds of bone in the skull. And so there's been, actually going all the way back to De Vere in the 1930s, there have been ideas about how the interaction of the chondrocranium, or the part of the skull that's derived from cartilage, and the dermatocranium, or the part of the skull that forms by intermembranous ossification, can be relevant to understanding how the skull forms. So we've worked on this now in a number of different mutants, and the overarching conclusion that I just want to show here is that when you perturb the growth of the chondrocranium, particularly the cranial base, you get the same shape effect no matter how you do it. Whether you affect the extracellular matrix of the synchondroses, or the growth plates of the skull, or whether you affect the rate of proliferation, or whether you induce bridging so that the growth plates fuse, you basically get a very similar set of phenotypic effects. It seems that the way in which the chondrocranium grows is a major determinant of the overall shape of the skull. And in this case, when you look at how much of the variation in the overall shape of the skull in humans and in mice is explained by an estimate of chondrocranial size, it's very large. It's about 7% in both humans and also in mice. So clearly, the growth of the chondrocranium is an important driver for craniofacial shape. Okay. So the third example I wanted to talk about is allometry and the integrated effects of size and shape. So I think allometry is fairly familiar to most people, most evolutionary biologists. I keep having to explain allometry when I work with people in the biomedical community because it's not a familiar concept there. But allometry, of course, is simply the shape effects of size. And we're used to seeing this, seeing the fact that if you look at people around us, there are proportions and shapes that correspond to different sizes. I could have put up a picture of Fred here, but I didn't have a picture of Fred in the same context. But if you take a tall person, this is me a few years ago, and this is my friend Dennis Ulatos, and Dennis looks like he's a real brute here with this huge head, and he looks completely out of proportion, and that's of course because Dennis is much shorter than I am, and now he looks perfectly normal, and this gangly guy with the small head looks kind of unusual by comparison. But this is simply a consequence of the fact that there's an expected relationship between size and shape in morphology, and we certainly see this in the skull. So this is just a large sample of inbred mouse strains, and as well as different mouse mutants showing this relationship between the size of the skull and the shape of the skull, and this is what a 3D morph of that regression looks like. See the same thing in the human face, but one thing that has surprised me a little bit is that allometry explains a much smaller proportion of variation in the human face than it does in the mouse skull. I have some ideas as to why that might be the case, but maybe that's a topic for later. So the thing about allometry is that it's much discussed and poorly understood. We know that there are shape correlates of size, and I think we have some idea that this occurs because tissues, structures respond differentially to underlying determinants of growth. But actually, this isn't well understood in most contexts. It's better understood in insects than it is in vertebrates. And so to try and get at this question a little bit, and the particular hypothesis we're pursuing here is the relationship between the regulation of the growth of the brain and the rest of the skull as a determinant of craniofacial shape. We took mice that were deficient in growth hormone and treated them at different ages with growth hormone to see how that would affect the eventual shape that you got. The idea being that if the skull and the brain are differentially responding to growth hormone at different times, you're going to get a shape that's going to be dependent on the age at which you administer the growth hormone. The alternative hypothesis is that it doesn't matter when you administer the growth hormone, they go through the same shape transformation and then they catch up, which incidentally would be the assumption that physicians make when they give a kid growth hormone. I think the fact that growth hormone is doing different things at different ages doesn't often enter into that equation, but plainly it does. So this is an experiment with mice showing that the shape effect that you get depends very strongly on the age at which you deliver the growth hormone. And the other interesting wrinkle in this is that you get the same sorts of shape effects whether you up or down regulate growth via growth hormone or an environmental perturbation. So here's my work that we've done in rats, showing the effects of pre and postnatal protein calorie malnutrition on rats, and you get roughly the same kind of shape effect as you see in mice with the growth hormone model, and you also get very similar shape effects in teeth. And here in a mouse model is the effect of protein malnutrition in prenatal growth, and also we see that the effect of the stress effect, the effect of reducing the protein content in the diet, produces a very similar kind of shape effect. So this is what a stressed mouse would look like compared to the control. And this is ontogenic allometry. And you can see here the similarity between these two axes of variation. And similar things happen in humans. So in the Tanzanian study, one of the things that we've done, in addition to working with craniofacial shape, is also to look at growth of the children we've been working with. And this came about because as we were working with this large sample of kids in Tanzania, we were interacting with both the kids, the parents, and the teachers. And one of the questions that you often get to is, well, what do you expect to see out of this research that would benefit you? And the consistent message that we got was, well, we're worried that our kids are malnourished, and we would like to know how our kids are doing, and we also want to know whether there's variation in how our kids are doing between different communities, because that information is potentially very important. So we've studied growth faltering in this population, and what we found is that for both girls and boys, there's a very significant amount of growth faltering, and interestingly, it's particularly severe at later ages. So in the school-age population, they're sort of close to the WHO standard. So this green line here is the CDC growth standard. And they deviate increasingly from that standard as they get older. And the boys do so more so than the girls. So it's not the case, at least in northwestern Tanzania, that the girls are less well off than the boys. It's actually the opposite, and we don't know why that is. And interestingly, there is a craniofacial shape effect of growth faltering. So because we have, for these kids, we have genetic data, we have 3D facial shape, and we also have information about their growth. And the growth faltering effect looks very much like allometry. This is allometry here, and this is the growth faltering effect. And Joanne Cole recently, in Rich's lab, calculated the environmental variances for both stature and for craniofacial shape. And I'll show you the results here for craniofacial shape because it's interesting. It's a completely different trait than stature. And what we've been able to show is that there is community-level variation in the overall growth outcomes. So there's variation in how much growth faltering there is in different communities, and we can explain some of that. But that variation is due, or at least is closely related, to the environmental variance. So the correlation between the environmental variance from the first three PCs and the growth faltering, or the PC that captures growth faltering from the anthropometric data, is 0.63. And this is at a community level where there are many different sources of noise in these data. So to get a correlation that is that high must mean that the growth faltering is due to environmental effects, which of course we're not surprised by, but that has not previously been documented. Okay. So the take-home message then from these three examples is that developmental processes drive integration and morphology, and understanding these patterns of integration and how they structure the genotype-phenotype map is critically important for understanding complex traits, not just in the face, but you can make similar arguments about virtually any complex trait. And the other conclusion, and this is what comes out of the last example that I gave you, is that often genetic and environmental perturbations are flowing through the same developmental processes. And so you would expect to see the same covariance structure from genetic and environmental effects. And this is, of course, known from, for example, Jim Sherwood's work in the early 80s. But I think we can start pinning that down to the developmental underpinnings of integration. Okay, how am I doing for time? Am I okay? Okay. So the second part of my talk, and this is much, much shorter, is about robustness. And robustness is the tendency for developmental systems to produce consistent phenotypes in the face of both genetic and environmental perturbation. And of course, there are two main topics within which we talk about robustness: canalization and developmental stability. And of course, canalization is, as a concept, is attributed to Conrad Waddington, but also to Smalhausen. I think that's less well known. Waddington had this concept of the epigenetic landscape. And to me, the most important thing about the epigenetic landscape is that it captures this idea that mutations can not just affect the mean, but also the variance of a trait. It captures that directly as a visual metaphor. And this is, I think, what these pictures show. So there are many ways in which you can produce variation in canalization. I'm not going to do a review of that. I'm just saying this because I don't want you to think that what I'm going to talk about is the be-all and end-all explanation for canalization effects, because there are many ways to do this. But we've worked on the relationship between nonlinearities in developmental processes and canalization and epistatic interactions. And to see how that might work, this is a toy model that we actually put into a grant application that worked. It convinced the reviewers. So imagine that you have some nonlinear relationship between a developmental process and a phenotypic outcome. And if that process is nonlinear, all you need is to have a nonlinearity in order for additive variation in the developmental process to produce non-additive variation, dominance effect in this case, on the phenotypic outcome. So that's a form of canalization. And the consequence of this is the phenomenon of both dominance, but also variance heterogeneity. Because for the heterozygote in particular, the same amount of developmental variation in whatever this factor is going to produce a larger amount of phenotypic variation depending on where you are here on the curve. Okay? If you have a two-locus model, you get both dominance, variance heterogeneity, and also epistasis. And if you have canalization here within the sort of a plateau within the curve, who knows if this actually happens in nature, but one could predict that selection might produce shapes like this, you would expect to see canalization within the range of the heterozygotes, and then increasing dominance and epistatic effects as you move further away. So go back to, think back to the sonic hedgehog example that I talked about earlier, where you have this relationship between sonic hedgehog expression and craniofacial shape. And the thing that I want to draw your attention to is that the curve that we got has sort of a hill function-like slope. And that's a bit arbitrary to define because it's difficult to relate this axis to this axis. But if you forget that complication for the moment, just bear with me and accept it when I say that there is a nonlinear relationship here between sonic hedgehog expression and craniofacial shape. And you can see that better here, actually, where we've better quantified the variation in the upper part of the curve. But clearly, the upper part of the curve is nonlinear. So you would predict, then, that if you go up and down this curve, you're going to get lots of phenotypic variation here, and less here, and less here, right, for the same amount of variation as sonic hedgehog. And so we've actually been working on a number of different ways to test that. And I'll just talk very briefly about one of them here that's unpublished. So this is work with B9D1 mice, collaboration with Rohlf and also with Jeremy Ryder. B9D1 mice have reduced sensitivity to sonic hedgehog expression. So it would be as if you were changing the amount of sonic hedgehog. So you're sliding down the curve. And what you get is a shape change that is exactly what you would predict based on changing the sonic hedgehog. You get a narrower face because of the reduction in sonic hedgehog, basically the reduction of the effectiveness of the sonic hedgehog signaling in the face. But you also get this substantial and significant increase in the phenotypic variance because of sliding down that curve. And this is the same thing just at a later developmental time point. And just to show you that this phenomenon likely occurs in many other systems, and it's probably quite ubiquitous. So this is another system in which we've been starting to work. So this is work by Rebecca Green, a postdoc in my lab, and also Jennifer Fish, collaboration with Rohlf and Trevor Williams. And so we've changed FGF8 expression, in this case via a genetic model. And what you see is that there is this interesting nonlinear relationship here between gene expression and, in this case, the second principal component of craniofacial shape. And depending on where you are on that curve, you're going to get a change in variance. So these genotypes here that are on this steep part of the curve also see an increase in phenotypic variance, just as the model would predict. Okay. So does this mean then that nonlinearities are an important contributor to robustness? And I should point out that actually isn't my original idea. The idea, I think, I found it initially in a paper by by Fred Nyhout and Chris Klingenberg from 1999 in the context of fluctuating asymmetry, but we've applied it here in the context of canalization. So if nonlinearities contribute to the genetics of complex morphology, we can make some predictions about that. So first of all, there should be significant genetic variation for variance heterogeneity because genetic variation is going to move you up and down these nonlinear curves, and that will produce heritable variation in canalization. Also, the genetic basis for the canalization of the trait should be similar to the genetic basis for the trait itself. So the nonlinearity is an intrinsic property of the system. It's not as if you got some canalizing genes out there and then genes determining the trait. It's the genes that are actually involved in the trait itself that are producing the variation in canalization because of the architecture through which variation is working. And the third prediction is that if this is ubiquitous, if highly nonlinear developmental processes contribute significantly to the variation of the trait, the genetic variation for that trait should exhibit a lot of non-additive variation. Because again, if you're going up and down the curve, you're going to produce dominance effects and epistatic interactions. And so I'll quickly go through the tests of these predictions. So this is variation in fluctuating asymmetry and in non-genetic variance, or the phenotypic variance that corresponds to a particular genotype in the collaborative cross mice and all their F1 crosses. This is work done by Paula Gonzalez in my lab, or when she was in my lab, and it shows that there's significant variation in both canalization and developmental stability among the crosses of the collaborative cross. And just curiosity, so this is what the first principal components of that variation, of the pooled fluctuating asymmetry or the pooled within genotype individual variance look like. Okay. So that's prediction number one. Prediction number one is definitely true, and we've demonstrated here in mice. There are other cases in which there's heritable variation for robustness shown. So the second prediction is that the genes that determine variation in the trait should be the same ones that are involved in canalization. There should be a high degree of overlap between genetic influences on canalization and the genetic influences on the trait itself. And so we go back now to our facial shape GWAS study, and also work that's currently ongoing on the genetics of craniofacial shape in the collaborative cross mice. So this is the diversity outcross group of the collaborative cross mice. And we see the same effect in both mice here and in humans, where there is a significant correlation. It's not terribly high, but there's lots of noise in these data. There's a significant correlation between the effect of a SNP on the phenotypic mean and the effect of a SNP on the phenotypic variance. And I actually didn't expect to find particularly high correlations here because most of these SNPs are actually probably noise. They're probably not really associated with variation. We picked a cutoff of p times 10 to the negative 5 for the ones that we selected. But still, there is a tendency for the same genes that are involved in the trait to also be involved in the phenotypic variance. And this is what the effect sizes look like for both the phenotypic variance here and for the mean as well. And we've also looked at additive versus non-additive variation in the collaborative cross more directly. And so the collaborative cross, as I told you earlier, is this massive recombinant inbred strain study of 8 founder strains that have been crossed in all possible combinations. And we've estimated the amount, and this is work by Chris Percival, mainly in my lab. So we've estimated the amount of additive variation and also the amount of non-additive variation that you see in the different parts of the skull. And so what Chris estimated is that for the overall face, about half of the heritable variation is additive, and a quarter is non-additive. So how much did we expect to find? I'm not sure. But if a quarter or so of the heritable variation is non-additive, then that's actually a big problem for genome-wide association studies of craniofacial shape. It means that there's a lot of variation there that the standard models are not going to be able to detect. And if you take into account the fact that most nonlinear processes or non-additive processes are actually going to produce a combination of additive and non-additive variation to begin with, that means that there's a lot of nonlinear type of stuff or other sources of non-additive variation that's going on here. Okay. And again, we found the same thing in the analysis that Philip Middoroker did in the same ways. And here's a specific example that Chris has been working on. So this is a series of mouse mutants for genes in the Sprouty pathway. Genes in the sprouting pathway are involved in the tyrosine kinase pathway, so they influence receptor tyrosine kinase signaling. And of course, as you probably know, that's a very deeply embedded pathway. It affects lots of different things. But still, you get fairly distinctive craniofacial shape effects as a result of mutations in this pathway. But the interesting thing that Chris has found is that the effects that you get depend very strongly on the genetic background. So you put the same mutation on a different background, and not only do you get a change, sort of a background dependency, but in some cases you get exactly the opposite effect, where an effect that's totally in a different direction, which I would interpret to mean that because this is a deeply embedded pathway, it means that the particular processes that are being most influenced in each combination is going to depend strongly on the pattern of epistatic interactions that are surrounding that effect in each mutant. And so this is a clear example of very strong non-additive interaction effects in development. Okay. And actually, I'll skip over that. So just in summary then, I think we can all agree that canalization emerges from developmental architecture. At least we've shown that in the case of the nonlinearity effect. That the developmental genetic basis for canalization is unlikely to be distinct from the developmental genetic basis for the variation in the trait itself. And that this process, or this type of phenomenon, nonlinearities in developmental process to phenotypic outcome relations, is an important potential source of genetic variation in canalization or in robustness. So what does all this mean for complex traits in general? So I hope I've convinced you anyway that the big thorny problem of trying to understand variation in the shape of the skull is well approached from the middle out rather than from the genotype to the phenotype. And if you make hypotheses at the developmental process level, and then make predictions about how that's going to affect the structure of phenotypic variation, you can get at insights into the developmental basis of variation in complex traits that are different, but more integrative and larger than that you would often get from a summary of individual genetic effects from genome-wide association study, for example. And I think the same approach is important for understanding complex traits in personalized medicine. Most diseases, in fact, most of the diseases that we're interested in are complex traits. So diseases like diabetes, obesity, highly polygenic, highly complex. And trying to understand, or trying to understand their developmental genetics in ways that jump directly from genetic variation to phenotypic variation is probably not realistic. We should be spending more time trying to explain these phenomena at multiple levels instead of only going directly from the genotype to the phenotype. So focusing more on the stuff in the middle. And if we think back to the stature example that I showed you earlier, so long bone growth is a major determinant of stature, as is the growth of the vertebral bodies. So what their network analysis showed is that growth of cartilage and chondrocyte proliferation is an important process in terms of determining stature. Well, if you know something about the biology of stature, was there ever another alternative? Was there ever an alternative hypothesis to this idea that growth at the growth plates is an important determinant of stature? And if we had started there, as opposed to getting there via a genome-wide association study of 250,000 individuals at how many $1 million, I have no idea, we'd probably be further ahead now in terms of understanding the developmental genetics of stature. Of course, one problem is there's a lot in the middle. So if you're advocating this sort of middle-out approach, where do you start? There's so many levels of explanation to try and choose from developmental systems. And I don't have a simple answer to that. I think it's highly context specific. But what I can tell you in terms of pointing towards the future is that the integration of imaging and morphometric methods that are applied to imaging data will play an important role in this, because I think that the answers will lie much more in approaches that emphasize combinatorial methods, machine learning-based methods that integrate data of different kinds. The same kinds of methods that are being used now for big data analyses are going to be much more productive as ways to understand the developmental genetics of complex traits than the standard SNP-by-SNP multiple univariate test model that is currently being applied. For that to be successful, not only will morphometrics be important, but morphometrics in a way points the way. Because right now, for example, if you do a genome-wide association study using a trait like the shape of the face, which of course is a highly complex trait, best studied using multivariate methods. You end up doing these sequential univariate tests on the genome side to test or to associate with multivariate measures on the phenotypic side. It would make much more sense to have integrated models in which we're doing multivariate statistics on both sides of the equation, or doing new things actually that involve implementing machine learning approaches that would integrate imaging and morphometric methods with genomic, proteomic, and various kinds of data that all come from the middle. And I think with that, I would like to thank my lab and my collaborators, and all of whom make it a real joy to be working in this field. And of course, without whom none of this work would really be possible. And thank you very much for your attention.